Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs 2016 Applied Biology and Methodology. If you are new to my channel, please do like, share and subscribe. Now coming to the year June 2016. So the first question. Which one of the following will be observed when auxin to cytokinin ratio is increased in culture medium during organogenesis from tobacco pith callus? So the answer here will be, it will not be adventitious roots will form, but there will be no shoot formation, because the auxin to cytokinin ratio is increased in the culture medium during organogenesis. So there will be no shoot formation. Now next question. In an effort to produce gene knockout mice, a gene targeted homologous recombination was tried with the exogenous DNA containing neomycin resistance gene and TKHSV thymidine kinase gene. Confer sensitivity to the cytotoxic nucleotide analog ganciclovir. If the neomycin resistance gene was inserted within the target gene in the exogenous DNA, and considering that both homologous and non-homologous recombination is taking place, which one of the following statement is not correct about the possible outcome of the experiment? So this is the experiment of homologous recombination method to produce gene knockout mice. I have already explained this in one of my previous slides, I think last year, 2015. This is 2016 question paper. I have done it in 2015 question paper. You can refer it there. I have given diagram also. It is the question is asked based on that diagram which I have given. I am not explaining it here. I will just see to the options. Cells with non-homologous insertion will be sensitive to ganciclovir. That is correct. So as per the question, it is which one of the following statement is not correct? They have asked not correct about the possible outcome of the experiment. So this is one. So option 1 is one of the possible outcome of the experiment. Then next one. Non-recombinant cells will be sensitive towards G418 and resistant to ganciclovir. That is also correct. Then homologous recombination will ensure that cells will be resistant to both ganciclovir and G418. That is also correct. That also I have given in the figure which I told you earlier in 2015 year. Then homologous recombinants will grow in G418 containing media. That is correct. But will be sensitive towards ganciclovir. They will be resistant towards ganciclovir. Answer is option 4. Then next one. In a typical gene cloning experiment, by mistake, a researcher introduced the DNA of interest within ampicillin resistant gene instead of the lac-Z gene. The competent cells were allowed to take up the plasmid and then plated in a media containing ampicillin, X-gal and IPTG and subjected to blue-white screening. Considering all plasmids were recombinant, which one of the following statement correctly describes the outcome of the experiment? So for cloning in a gene cloning experiment, what did the researcher do? He introduced the DNA into the ampicillin resistant gene instead of lac-Z gene. Then he prepared the competent cells and transformed the competent cells with this plasmid. Then he plated the competent cells which have taken up the plasmid onto a medium containing ampicillin, X-gal and IPTG and subjected to blue-white screening. Now considering all plasmids were recombinant. Now all the plasmids are recombinant as per the question. Which one of the following statement correctly described the outcome of the experiment? Remember, he has introduced the gene in the ampicillin resistance gene. So naturally none of the plasmid will grow in this particular medium after plating because it contains ampicillin. So the answer here will be the bacteria which took up the plasmids would not grow. So answer will be option 2. The next one. Radioimmunoassay can be employed for the detection of insulin in blood plasma. I am not going into the details of how radioimmunoassay is conducted. I hope you all know it. For this, I125 labeled insulin is mixed and allowed to bind with a known concentration of anti-insulin antibody. A known volume of patient's blood plasma is then added to the conjugate and allowed to compete with the antigen binding sites of antibody. The bound antigen is then separated from unbound ones and the radioactivity of free antigen are then measured by a gamma counter. Following are some of the statements made about this assay. So first, I125 labeled insulin is mixed and allowed to bind with non-concentration of anti-insulin antibody. Then the patient's blood is taken and is allowed to compete with the antigen binding sites of the antibody. The bound antigen is then separated from unbound ones and radioactivity of the free antigen is measured via a gamma counter. So here the patient's blood will be one normal and one diabetic. Okay, because it is insulin. So the normal patient as well as diabetic patient, they have given the statements for both normal patient and diabetic patient. We have to see. Now what did they do in the case of the normal patient? So antigen, that is labeled insulin, it was added to specific antibody, antigen antibody complexes were formed. Okay. Then after that insulin from the normal person, that is unlabeled antigen. It is from the normal person was added to the complex. The unlabeled antigen competes with the labeled antigen for binding to antibody. The unlabeled antigen, that is insulin from the normal person, will compete with the labeled insulin for binding to the antibody. Now the amount of insulin is more in the normal patient. So the unlabeled insulin will displace the labeled insulin from the antibody and it will bind over there. So the amount of radioactive labeled insulin will be more, or it will be the free fraction, unbound fraction. So the amount of, and it will give this labeled insulin, the amount will be more, so it will give more signal in the gamma counter when measured by using a gamma counter. So the amount of unlabeled insulin bound to antibody is more than that of labeled unbound insulin. Okay, the amount of unlabeled insulin bound to antibody is more than the labeled unbound insulin, that is labeled unbound insulin in the free fraction. So what is the first statement that they have given here? The ratio of radioactive count for unbound antigen. Unbound antigen is unlabeled insulin to the bound one, that is the one which is bound to your antibody, that is the insulin from the patient which is bound to the antibody is more at the end of the reaction. So the ratio of radioactive count for unbound antigen, of course it will be more at the end of reaction because the patient's insulin will compete with the labeled insulin and it will bind to the antibody. So the unbound antigen, that is the labeled antigen, it will be in the free fraction and it will give more value for your gamma counter. Now coming for the diabetic patient. So diabetic patient also the same experiment was conducted. So I will tell you how. So antigen, that is labeled insulin, is added to specific antibody. Antigen antibody complexes are formed and insulin from the diabetic patient, that is unlabeled antigen, is added to the complex. The unlabeled antigen competes with the labeled antigen for the binding sites for binding to antibody. As the amount of insulin is less in the diabetic patient, The unlabeled insulin cannot displace the labeled insulin. Okay. As the amount of insulin is less in diabetic patient, the unlabeled insulin cannot displace the labeled insulin. That is, the amount of unlabeled insulin bound to antibody is less than that of labeled unbound insulin. So for diabetic patient, the radioactive count for free antigen will be less than that for a normal individual. That is your statement 3. So which of the above statements are true? 1 and 3 are true. In case of 3, the free antigen is less. So it will give only very less signal in the gamma counter. Okay. So that is the in case of radioimmunoassay. Now coming to the next. A researcher wants to obtain complete chemical information, that is head groups and fatty acids of phospholipids from liver tissues. Phospholipids have fatty acids of different length and unsaturation, and also the head groups are of different chemistries. Which of the following combination of techniques would provide complete chemical description of phospholipids? So it is naturally TLC, thin layer chromatography, and gas chromatography that is going to provide you the required information. Answer is option